Thank you very much. My name is Mathieu Boudreau, and today I'll be giving you a tutorial on doing qMT using our software, qMRLab. So I split up this talk into three parts. First, I'll give you a brief introduction to the qMRLab software and a little bit of history. Then I'll dive into the tutorial portion of the talk. And finally, I'll let you know on some of the cutting edge work we've been doing with qMRLab and the greater qMRLab ecospace. Say. Got nothing to declare. So all the content that I'll be made for this talk will be shared on this GitHub repository. You can come here and take a look at the slides. I've written up the tutorial as a step-by-step document, and we'll also provide a live video. And in a few weeks, we'll also be publishing a blog post on our website, which will cover some qMT theory and have some interactive tutorials as well. And this will be done by one of Nicola's current students, Swan. So our software is qMRLab. Our motto is quantitative MRI under one umbrella. And this is a MATLAB-based software that is about, let's say, 7 years old now. It's based on two major publications. So it started off as being qMT-Lab. So Jean-François Cambana published this in 2015 and created this open source software that encompassed data simulation, analysis, and visualization of a lot of qMT methods that were available in the literature, but brought them all under one software. And then after that, we continued adding other tools such as T1, B0, B1, which are needed for some qMT methods, and then extend this to a lot of other qMT techniques. And this led to the JOS paper for qMRLab that was written by Aga. So there's a lot of stuff that you can do with qMRLab. This visualization summarizes it well, I think. So like I said, quantitative qMRLab, our motto is quantitative MRI under one umbrella. So we cover some relaxometry, diffusion imaging, QSM, field mappings like B0 and B1 and qMT. You can fit, simulate, and visualize the data. And now Aga's working on extending this software from scanner to publication. And I'll talk about that at the end of the talk. So there's a lot of MT methods available in qMRLab. So we have MTR, MTSAT, and qMT, sorry. But for this talk, I'll just be going through this little pipeline here. So we'll just be talking about SPGR-based qMT, so spoiled gradient echo. And we'll just be using the default tool, so which is the default fitting model, which is the Sun Pike. So as some of you might know, qMT, especially done using SPGR, requires some additional maps, so B0, B1, and T1, but our demo includes these already fitted, so I won't cover how to produce these maps. You can produce them either with our software or with another software, and then just load them. So I'll jump into the tutorial. First, you'll want to download the qMRLab software. And this can be done a few different ways. You can go on our website, qMRLab.org, and download one of the release versions. You can go on our GitHub page and download the current development version or one of the tagged versions. But if you're following the software at home later on, I'd recommend that you go and check out the tagged release for ISMRM 22 and just download this version. And this will be integrated into the main repository a little bit later on. To run qMRLab, you'll need either MATLAB or Octave. Unfortunately, the graphical user interface is only available on the MATLAB version. You can do the command line tools using both. So for this tutorial, I'll assume that you have MATLAB already installed on your computer. So I'll give you a brief overview of the qMRLab interface. On the left panel here is the main window for qMRLab. So here is where you'll select the different methods that you want to do, if it's qMT, T1, B0. You can load your data here, visualize it, get some statistics on it. This is where you'll start fitting the data or saving and loading some results. And you can also select some of the simulators as well. On the right panel is the options graphical user interface. So for each of the methods that you've selected, there will be some respective methods such as the protocol parameters. So these are all the default versions that are loaded when you load a qMT-SPGR. Timing tables, some of the fitting parameters such as the lower and upper bounds for some parameters, and additional model options such as here the MT pulses and model line shapes and fitting constraints. So for this talk, I thought I'd give you, I guess, the workflow that I would recommend a brand new user that might be, one, not familiar with qMRLab, and two, that might not be, it might be the first time that they're doing quantitative MT. So I think before you dive into, fitting a data set and looking at the results, it's important to understand what you're looking at and if it's, if the data was acquired correctly or if there might be some errors. And so I find simulations are very useful to get some of this intuition, and it's a good way to explore how some potential errors or sources of errors of your data prior to, or if you've gotten your data, and specifically if you're designing your pulse sequence, then this is good, or protocol, this is a good way to test out your protocol before you go to the scanner. So first off, I'll just show you how to fit and or simulate and then fit a single voxel of data using our software. So for this, you'll want to go to the left pane of the main window, and highlighted in red is a button called Single Voxel Curve. If you click this, a new window will open, and the two main windows will appear, will still be in your background, but I've removed them here for simplicity. So this is the new window that opens, and you'll have some options that you can select. So first off is what level of noise that you want to simulate with your data. How do you want to simulate your data? Some methods. For qMT here we have what we call analytical equation. There's also the block simulator, which will simulate several iterations of the TRs. And also you can select what your tissue or species parameters are. So for this demo, the first time you're doing a demo, I'd suggest going to a very high SNR. You want to simulate what's essentially a noiseless signal, an ideal signal. So here I've selected or I've typed in 1,000. After that, you can change the parameters if you want. For now, we'll keep these as default, and then we'll vary them a little bit after so that we can see how the Z-curve changes. But next, you can take a look at the options window. This is where if you wanted to simulate, for a different protocol, you could do so. Note that if you change anything in the protocol pane, and also I think in the MT pulse pane, that you might get an alert saying that the SF table is not is not cached for this protocol. Essentially, you can pre-calculate a dictionary of some intermediate values, which we call the SF table, once you've set up your protocol, and that'll speed up the fitting. But if you're just using the default, you don't need to worry about doing the SF table. And if you're just doing single voxel, then you can just ignore this warning. It's just if you're processing a large data set that I compute another SF table, which takes a little bit of time. So go back to your other window. You click Update Fit. Because we've selected the analytical equation, it should be immediately shown. And here you'll see the normalized signal values for each of the protocol points that we require, so each of the offsets and each of the flip angles. And this is what we call the Z spectrum in qMT. And above there, you'll see the table, and the table will show you what the input parameters were and then what the fitted parameters were. And so that'll give you an estimate of how some parameters fit. So if you want to see how the curve changes for different parameters, here I selected F, which is the pool size ratio. It's related to the macromolecular content. So the higher the number is, the higher the macromolecular content will be. And then intuitively in MT, that means more saturation will occur, and so we should see more signal from the water pool go down from the exchange of MT. So if we lower this value, You could think of going from white matter to gray matter, so 0.16 to 0.5. You'll see the curves, there's less saturation going, the curves go up, and even lower, they go up, but they don't go up equally across all the offsets. And this is because there's still some on-resonance stuff going on when you get close to these values. If we reset to 0.16 and we have a higher micromolecular content, we'll see the curves go down. 0.25 and then 0.5, which was our upper bound limits for the fits, they land here. If I can go back and forth, you see how the curves change a little bit. Next, I'd recommend maybe running a block simulation of your sequence. And so this will ensure that for the pulse sequence that you've selected or the protocol that you selected, that you acquire enough preparatory pulses to reach a steady state, because for this qMT technique, there's an assumption when you're doing the fitting that you've achieved the steady state for all of these points, and it's not always a guarantee depending on your protocol values. So here in methods, you can select the red box and click block sim, and then click update fit. And so right now, what we're currently seeing is the results from the analytical equation, and if I move forward, we'll see the here it's going to take a little bit longer to do the simulation because it's running each of the TRs. Keep going. So right now what we're seeing was the results for the analytical equation, and then we'll see the results for the block equation. And for 600 preparatory MT pulses, we can see that it's approximately the same. So this is a good number for all of the pulses. There's slight variations here and there, but it's pretty close. If you go in the options pane, this is where you can change that number. So it's set to 600. So if you go really, really low, guaranteeing that you didn't achieve steady state, let's say like 10, and you go back and you click update fit, you'll see that the fits aren't working, and the pulse, the data points don't look like the spectrum that we would expect for this protocol. Increasing this to 20, we can see where the trend goes. So steady state's coming, 50 is looking close, but like this could be, this almost looks like, except for the first data point of the off-resonant frequency, something that I could imagine somebody would get from a first acquisition and wonder like why are my fits going so bad. The Z spectrum kind of looks like what I would expect, but it's because you haven't reached the steady state. And then if you go to 100, you're almost there, and the fit values are getting fairly closer. 0.16 to 0.18, it's almost indistinguishable, but you'd probably still want a higher number if you want really accurate values. So there are other options that I wanted to highlight. There's a lot of options that you could choose, but I want to highlight these two. First is the View Protocol button. And so if you look at the timing table, there's all these abbreviation TM, TTS. You won't intuitively know what these mean. If you click on Plot Protocol, it'll open a window, and it'll show you a simplified version of the pulse sequence diagram and what each of those parameters mean. And you'll also see the empty pulse shape, and if you change the empty pulse shape, you can see how that changes as well. And on top, you'll see the empty parameters, so the flip angles that you've acquired. Here we just have 2, and then the offset frequencies. The other thing I wanted to highlight is line shape. So if it's your first time doing qMT, I would just want to point out that it's important that you select the correct line shape for what you're acquiring. So when you're acquiring humans, it's assumed that the absorption line shape is going to be from the restricted pool is going to be super-Lorentzian. If you're testing this first on a phantom, this will be the incorrect choice. So you'll want to select Gaussian, and you see how the Z curves are different for both. And so if you've acquired data on a phantom and agar phantom, but trying to fit it with super-Lorentzian, your values are going to be completely off. So that's another important thing to choose. Things like the model, you can explore that. There's some papers that compare the different fitting models. I won't go through that. So now that we've completed the demo, or the simulation part of the demo, I'll just show you how to download one of our data sets. And so each of our qMRL models come with, we've uploaded a demo data set that you can test the fits on and test the software on before you use your own software. This is also a good way to know how to format your data to upload to load it in our software as well. So these give good example for that. So each of the models will have a button here that says download example. You'll click that. It'll open a window telling you that it's going to download and create a folder. You click OK and then choose wherever you want to download that. You'll need internet access when you're doing this, and that'll, depending on how large the data set is, it might take a little bit of time, but the qMT is not too large. So you click open, and after a while, all of the parameters will be populated. So here, MT data and RO map are highlighted, are bold. These are because they're some of the required maps, required data for this model. And then the ones grayed out, B1, B0, and mask are optional. So the mask will just help speed up the processing of the whole volume. If you don't provide it a B0 map, it's just going to assume it's homogeneous and 0 everywhere. If you don't provide a B1 map, it's just going to assume it's homogeneous and 1, or the normalized 1 B1 everywhere. Once you've got data loaded, you'll want to visualize it and get maybe some intuition on if the data looks good. So here I've selected view for the B1 map. So I'll click here, and this is the first thing that you'll see show up. What's highlighted in red is going to be the mask overlaid on the data. You can deselect this by clicking this checkbox here, and that will remove it. Because this is a quantitative map, the default grayscale, is not really useful. So you can select Perula or Jet. I've selected Perula here. This looks a little bit closer to what we see in publications for B1 maps. And then you can, because it's still very noisy in the background, the mask doesn't remove the noise in this step. It'll remove it when it's doing the fit. You can change the lower and upper bound so that you're within just the human range. So here I've selected 0.7 to 1.2, and you can see our B1 map. Now, if you want to get some additional information, what does the data look like? You can click on the histogram button. That'll give you the curve. This is what it is for B1 map. If it was for the R1, R1 is 1 over T1. You should see two peaks, one for white matter, one for gray matter. And then for MT data, I don't think would be useful. If you want to look at a specific region in the brain, you can click on the select button next to cursor and click somewhere on the volume, or even drag it, and you'll see how the values change. Here it's close to 1. Once you've got all your data loaded, before fitting the whole volume, I'd recommend that you verify the fit for a single voxel to see if your acquisition looks correct in a region that you would expect it to be correct. So if you select a region of white matter, see if the parameters, the fits go along the data points, and make sure that the fitted parameters seem approximately appropriate. It'll be noisy, so it'll be there can be some variability. So here you can select cursor. And here I've clicked just a region of white matter. So this volume, sorry, I changed it. It's the R1 map, so the inverse of the T1 map. The value that I've selected is close to 1, so it's a T1 close to 1,000 milliseconds. It was acquired using VFA, so it's a little bit higher than what you would expect from inversion recovery, for example. But so I've selected this data point, clicked on the sanity check region. So where it says voxel fit, and that will fit the data for this point. And so here you'll see the curves. It's a little bit noisier than the simulations that we were doing. You'll see the fitted values. So my first impression here is like, okay, f.24, is a little bit high for white matter. It could be due to a lot of different things. It could be due to the noise. Maybe on the higher end of the offsets, it could be due to the R1 maps that are incorrect. So here's where you would go back in the simulations and see what contributes maybe the most for this, or like what curve also would match these parameters. So what you would have expected. If you want to compare this with another region, you can click on the button whole plot in order to compare voxels. So here I selected a region of gray matter, and then click voxel fit again. And here the old plots will be grayed out, and you'll see the new plots. So the pool size ratio for this region was lower, and so the curves go up. Now, once you've done all that and the data looks good, that fitting the entire data set will probably is the easiest part, really. So you'll go to fit data, everything's loaded, your protocol is correct, you've computed your SF table for your protocol if you've changed it. It'll ask you if you want to do parallel processing. If you're doing this on an old laptop, maybe say no for the first time that you're doing it in case it crashes your laptop, or it can slow down significantly. But if you can use parallel processing, then it'll accelerate your the fit of your entire data set. So here it'll fit the data. Imagine that time goes by, and then it's loaded. So here you've got the pool size ratio, the F that's shown up at first. It'll reset the color maps because it doesn't really it hasn't attached any color maps with the parameters yet. So change that to Perula, change the bound. Now it looks kind of good. You can change the source if you want to take a look at what some of the other maps that were fit, or the res norm. So here's T2F, for example. So here I've really only given you a tutorial on the graphical user interface. There's a lot of other options that you do, the command line interface, scripting. We provide some batch demos. So if you open the file qMT-SPGR batch, it'll provide you a line-by-line demo to essentially fit the data set that I just shown you. And so you can use this, for example, to do also simulations. So you could simulate, for example, like the example that I was saying earlier, simulate a data set that's using a Gaussian line shape, and then fit it with the superannuation line shape to see how, what type of errors you'd see, or fit it with one protocol, or with one method, and then simulate with one method and then fit it with another model. I could see this being useful also if you wanted to simulate MTSAT data or MTR data. You could do this in the qMT and then try to fit it in the other modules. So that's ends the tutorial part of the talk. I just wanted to briefly mention some work that's being done in our lab, primarily by Aga Karakuzu. So he's presented this work at this ISMRM, so it's Abstract 366. That was done yesterday, and it was also accepted as a paper in MRM. So his work focuses on what he calls qMR flow, so it's container-mediated and data-driven pipelines using the Nextflow software or framework. So this allows you, with the click of a button, to go from acquisition, converting this into ISMRD, then reconning it using Gadgetron, then converting into a bits format, doing some pre-processing, and then finally doing the processing qMRLab, all within different containers, and all with a Nextflow. Nextflow is platform agnostic, so you can do this locally, you can do this in the cloud or in a cluster. And the three key principles that he wrote this with is isolate, map, and use. And this is a demo that he did with MTSAT flow, and it produces workflow reports and quality control reports afterwards. So the qMR flow equation, if you will, is on top there, but that's only one part of his vision and Nicola's vision, which is from standard to publication. So it doesn't cover the pulse sequence part, and so he developed qMR PulseSec, which I think uses RT-Hawk And so that combined with qMRFlow is what he calls Venus, so venture-neutral sequences. And he's used this on a recent study on 3 different scanners. So he did this with MTR and MTSAT, and I think also T1, but I'll only be showing MTR and MTSAT. So he did this using the native pulse sequences on those scanners to acquire MTR and then MTSAT on 2 different vendors, and one of the vendors 2 different versions. Did this on a phantom and healthy participants, but the MT stuff was only done on the participants. And then repeated this using his venture-neutral pipeline. This was using the RT-Hawk interface to the scanners. And like you can see here for MTR, there's better inter-scanner reproducibility using his Venus pipeline. And same with MTSAT. Still a little bit maybe B1 inhomogeneity, but inter-scanner has good results. So I'd just like to thank current lab members, external contributors and users, everybody that's contributed to qMRLab in the past, and highlighted in bold here are just some of the really key developers that have contributed to the software or the algorithms over the years. And so thank you very much.